Traditional experimental drug screening is an exercise in scale. Physical laboratories require churning through thousands of chemical compounds to find a single viable candidate, a process that consumes significant funding and time. Molecular docking circumvents these physical limitations by simulating drug-to-target interactions digitally, allowing researchers to evaluate molecular bonds without pouring a single drop of liquid. Enter AutoDock Vina. Originating from the Scripps Research Institute, this is a free, open-source software engine designed to predict these binding affinities with high precision. By simulating these interactions digitally, virtual screening allows researchers to filter through massive compound libraries, prioritizing the most likely candidates before physical lab work begins. Before initiating a simulation, you must set up your environment. Step 1 is a local installation of AutoDock Vina, alongside a separate 3D visualization tool like Biomedical. Next, you need the raw biological data. This requires securing a high-quality 3D structure of your target protein and an isolated file for the specific ligand molecule you plan to test against it. Because Vina operates as a computational engine rather than a standard desktop application, you also need a basic familiarity with command line interfaces and plain text editors to run the software. Computational tools amplify human intent. If you fail to assemble and properly format these foundational files, the simulation will stall before it even begins. Preparing the protein structure is your first task. You have to clean up the source file by stripping away water molecules and irrelevant ions to ensure the structure represents an accurate biological state. Looking at your file directory, you must convert this cleaned protein file into a specific extension type, the PDB-QT format. This step is an absolute requirement for the Vina engine to read your data. Now you move to the ligand preparation phase. Here, you have to optimize the geometry of the molecule and accurately map its electrical charge. AutoDock Vina actively calculates how a ligand bends and flexes during interaction. Skipping this initial geometry optimization guarantees inaccurate results. Once the ligand is optimized, you run it through the exact same conversion process, generating a second PDB-QT file. In molecular modeling, your output quality is tied directly to the precision of your input. Careful preparation of these PDB-QT files is what makes a reliable prediction possible. Next, you define the docking search space. This involves targeting a specific pocket on the undulating surface of the receptor where the ligand will bind. You map the search space by drawing a 3D grid box over the target region, telling the software where to concentrate processing power. If expanded too far, you select irrelevant areas, increasing compute time without improving results. To feed these spatial boundaries to Vina, you create a plain text configuration file. This document holds the precise X, Y, and Z coordinates of your grid box, alongside your operational parameter settings. You must rigorously document these specific parameters. Tracking your configuration variables is required so the experiment can be reproduced and verified by others. By explicitly defining the search space and documenting every parameter, you move from a general simulation to a specific, reproducible study of a target pocket. You are now ready to initiate the docking simulation. You execute the run command directly through your terminal interface to start the engine. Behind the scenes, Vina utilizes multi-threaded processing. It distributes the computational workload across your machine's cores to rapidly process the calculations. Looking at this terminal output, you see a structured table. The engine has generated a list of potential binding poses, alongside a column for their corresponding binding affinity score. The core metric for success is straightforward. A more negative binding affinity score mathematically predicts a significantly stronger molecular bond. Because multi-threading speeds up the computations, you can quickly evaluate these outputs, isolating the most viable drug candidates using purely mathematical affinity metrics. Raw data only tells half the story. The final step is importing these output files into a visualizer like Biomedical to verify the mathematical data visually. Here you can see the final protein-ligand interaction, a small complex molecule locking into the designated pocket of the massive receptor. But this comes with a crucial scientific caveat. While computational predictions are highly accurate, they must always be validated by physical, laboratory-based evidence. You use this initial baseline score to begin an iterative process. You can return to your ligand file, adjust its chemical structure, and run the simulation again to achieve a better affinity score. When computational data guides laboratory testing, researchers can ignore thousands of low-probability compounds, focusing their resources on the handful of molecules with a high predicted affinity.